Great. So just going to pull this up on ISCB to make sure that we're live.
It's live.
Perfect. Yep. Great. So like I said, if anyone is here right now, we'll give it a few minutes, let a few more viewers stream in slowly, and then we can get to Raghu's presentation. Thank you all for everyone who's here right now. We're excited to have Raghu presenting today. And Raghu, I believe that we circulated the poster as well on our Twitter, so hopefully that will bring some more viewers as well. Yeah, the media team for ISCB has been fantastic with the quality of the posters that we're putting out. So it's a great help, of course.
They have some nice aesthetic qualities in designing the posters, so it came out really well.
Yep. Certainly. Great. So I think we can get started now. Hello, everyone. Thank you so much for attending this session for the ISCB-SC webinar. My name is Colin Small. I'm a member of the webinar team, and I'm very proud to be presenting Dr. Raghu Yanamali for his presentation today. Raghu is a current assistant professor in the Department of Bioinformatics at Sastra University in India, and his talk today will be on the computational studies of cellulases. So Dr. Yanamali, feel free to share your screen and start your presentation when you're ready.
Thank you, Colin, for the opportunity and for the nice introduction. Thank you so much. Let me just so are you able to see my screen? Let me pull on the pointer. Okay. Are you able to see my screen and the slide?
Yes, we're all set.
Yeah. Okay. Good morning, good evening, wherever you are. Thank you for joining in for this ISCB Student Council webinar. So today I'm going to talk about computational studies on cellulases. I work at the School of Chemical and Biotechnology, the Division of Bioinformatics under that school in Sastra University in Tanjavur in India. I'm more than happy to share what I have been doing for the past, I would say, like 10 years or so on this broad area of cellulases. So my area of research has been spanning multiple domains: structural bioinformatics, systems biology, machine learning, and then structural dynamics, both fine-grained and coarse-grained methods. And finally, we did some amount of functional analysis from sequence and also from structure. And before I go further, I want to thank my past and current lab members. All of them were involved at some point with the cellulase work, including Shankar, and my current lab members who have been working on these projects. Sri Vaishnavi has joined just recently on this particular work, but then Shashank and Yati have been doing a little bit more. But you will see these names coming frequently in the next few slides, and you know who they are. Okay, so most of my research has been funded by Sastra University, Department of Biotechnology, Government of India, and Indo-US Science and Technology Forum. And I thank my collaborators also at this point. Okay, so in '91, a movie came out called Driving Me Crazy, which was the entire premise was that an East German inventor tries to sell his invention of a car that runs on vegetables and produces no pollution. So it boils down to that the concept that if you have a renewable source of energy, and it is something that drives the entire world on many levels. So of course this movie was a slapstick comedy where he runs into one madcap after another, and then he ends up in some trouble. I'm not sure if most of you know about this movie, but I found this movie to be quite funny and related to what I do. Most of the time when we hear cellulases, we hear about either stonewash genes or detergents that use these enzymes to remove peeling. And one of the organisms that actually does most of the cellulase production is Trichoderma ricci, and this is a organism that's considered as a powerhouse of producing cellulases. Currently, the cellulose plant biomass conversion from lignocellulosic material to ethanol or biofuel has different generations. So one of the first initial generation was just fermenting glucose, that is from sugar, and to ethanol. The other one was converting the starch from corn, which is what most of the ethanol production in US is about, sugarcane mostly in Brazil. And the tomorrow's technology, which has been used in many cases in industry and but has not been implemented widely, is where you use a mixed sugars for converting into ethanol. So most of the enzymes that do the work of breaking down the polymers to mix to monomers are cellulases, which is a very big umbrella term. And so to schematically show, cellulose polymer has a non-reducing end and a reducing end, and the polymer is basically in a crystalline form in the plant and other naturally occurring sources. And what happens is, the first instance, the breakage that happens is the synergy between three enzymes. All three are cellulases, but this one is called endoglucanase, which breaks out a glycosidic bond that by opening up the chain for the next enzyme, cellobiohydrolase, that chews it up and gives out cellobiose, and finally beta glucosidase that ends up creating the monomers. So the one of the idea that had been in work for many years in the US and in some amount in other countries, where engineering a maize plant with the endoglucanase gene from the thermophilic organism called Acidothermus celluliticus, which was found in the hot springs in Yellowstone National Park. So the residual agricultural waste, when you do pre-treatment, it gives out the amorphous cellulose, which then is hydrolyzed in planta. That is, the hydrolysis starts happening, and because it has a very high temperature of optimal activity, the optimal activity happens only at certain temperature when it raises, and it doesn't get diminished, so there is a turnover rate also happens. So when I started this work, I asked the question like, okay, there is endoglucanases which work, and we are taking a thermophilic endoglucanase, but what is thermophilicity and what is happening in these endoglucanases? So basically the question was, what confers thermostability in endoglucanases? So it so happens that endoglucanases have diverse folds, and they end up doing diverse mechanisms. That is either a retaining mechanism, that is alpha beta fold, and beta jelly roll do the retaining mechanism, where this carbon orientation of the carbon is retained after the hydrolysis, or it's inverted in the case of alpha alpha 6 fold. So what we did was, we curated a data set of endoglucanases based on sequence identity cutoff, and then divided into different data sets, and then we kept those that were above 40 degrees Celsius as thermophiles or thermostable enzymes. So we did a lot of sequence-based analysis, and we did structure-based analysis, and then we found that certain amino acids in the thermophile were significant as an entire enzyme group, and also as the structural fold, and also as the mechanism compared to the mesophile. And at the same time, we found that these amino acids also have a preference where they want to be positioned in the structure, whether they are present as exposed, and also it will come in, and also depending on what type of interactions that they are having. So these darker gray cells indicate the amino acids or the interactions that are having a significance in that particular category. And similarly, this is what I was talking about, the positional preference, that whether they want to be in which part of the secondary structure and which part of the surface, buried intermediate exposed. So there are some patterns that we found based on just the sequence and the static structures. So when we did further analysis of the mesophile and thermophile comparison, we found that certain residues that are replaced in the mesophile as compared to the thermophile, there is a particular preference as to where they are located, and also what type of amino acids or what class of amino acids that they are replaced by. So here I'm just showing the alpha/beta 8-fold, where arginine and proline are replaced by polar amino acids, and leucine is replaced mostly by aromatic amino acids. So we had the same for the other two folds as well. So at the end, we came to the conclusion that thermostability in endoglucanases is fold specific, that the type of amino acids contributing to thermostability differ depending on the fold. There are charged residues that are important, but surface accessibility also plays a role. And there are some subtle interactions that actually have very minor changes, but they are kind of like the driving forces happening. But keeping in mind that all the data that we used are static structures and sequences, we know proteins are not static, they are dynamic. So definitely there must be some role of dynamics playing some kind of relationship in imparting thermophilicity. So the basic question was, what role does structural dynamics play in thermostability, especially in endoglucanases? And is there a relationship between the structural dynamics, the function, and the thermostability or thermophilicity of those proteins? So in this particular case, what we did was we took a case study as a proof of concept, that we took two proteins, one from thermophile and one from mesophile, which belong to the same fold, and they had a very high structural similarity, but not a very high sequence similarity. And the thermophile had more had more insertion regions that you can see in green here as compared to the mesophile. And this was very interesting for us. The reason is that at higher temperatures, protein tend to have very shorter loops, so that they become more rigid at higher temperatures. But that was not the case for this particular thermophile. So we thought, okay, this could be a good case study to understand what is happening. And these two residues here, one in orange is the nucleophile, and the one in blue is the acid-base donor. They are the key active site residues that play a role in the function. So here's the nucleophile and the acid-base donor of the thermophile and the mesophile, shown in black and gray respectively, and the interaction maps of the mesophile and thermophile in terms of the substrates that they bind. So they bind to cellobiose and cellotriose. So what we used was a very simplistic method that you can call, because nowadays there is GPU-based systems are available and you can do long time scale simulations, but we wanted to see whether we are able to capture the same long time simulations in using a elastic network models. And so a coarse grain approach that was proposed way long back, which is basically decomposing the dynamics into different modes, basically the slowest mode and the fastest mode. And there are two elastic network models, Gaussian network model and anisotropic network model, which we use to study the dynamics. So basically, elastic network model, to give you a background, is nothing but, sorry, I'll just do a little bit, one slide back. Yeah. Okay, so elastic network models are nothing but you're taking the entire amino acid as one entity. So each C alpha represents an amino acid, and if they are within a particular cutoff, that is 7 angstrom in distance, you connect them with a spring. So basically, when you take a protein, you take the backbone, which is the C alpha, and the backbone is showing you the main peptide bond that they have. But then what you see here is the gray bonds that the springs are being connected with it. So you get two types of modes. One is the slowest mode, the other one is the fastest mode. The slowest modes are associated with the biological function of the protein, and the fastest modes tell you which residues are highly fluctuating and they are kinetically hot. So what we saw with the first slowest mode that you see here, on the top is the thermophile, and on the bottom is the mesophile. And the thermophile, they are colored by red being the highly flexible regions, and blue being the least flexible, and green being the intermediate. There is an imaginary axis that you can see or you can plot here, and this has, even though it is one domain, there is an opposite correlation happening between these two domains in both thermophile and mesophile. And this is in the first slowest mode. And in the second slowest mode, we do see another type of a correlation happening within the same domain. So in the end, using this, there are other data that I'm not showing due to time constraint. So what we found was the loops that are facing the substrate binding site of the proteins, they are the most mobile in the slowest mode, which means they are involved in the function, of course. But then there is some kind of coupled and uncoupled dynamics happening between the nucleophile and the acid-base donor in the thermophile as compared to the mesophile. And in certain cases, that is in the thermophile, there were some large dynamic domains that we could found which we didn't see in the mesophile. So definitely there are some dynamic based differentiation that is happening, which we found correlating with whether it's the thermophile or whether it's the mesophile. But all these endoglucanases, as I showed earlier, they work on amorphous cellulose fragments, meaning that what you see here is the crystalline cellulose, and this cellulose strand that is pulled out from the crystalline part, that is now in the amorphous part, and that then the cellulase that binds to the polymer has a carbohydrate binding module which is connected by a linker. This is very, very flexible. You generally don't see an electron density in the structures. And so amorphous cellulose, what gets pulled out and gets chewed upon, is relatively easier to digest, but the crystalline form is not easy. So the crystalline form of cellulose is basically highly ordered, and 40 to 50% of the cellulose that's present in the biomass is crystalline in nature, basically the 1-beta form. So this is the top view of the cellulose looking from top, and this is the side view. You can see that it's quite planar, and the only thing that's sticking out on the top are the hydrogens from those oxygens and the carbons that are in the sugars. So it so happened that about 20 years ago, the cellulases were found that can act on crystalline form of the polymers. And these, while at that time they didn't know these are having an action, they knew that they were binding to chitin. So they called them as chitin binding proteins. Later on, they changed the nomenclature to Lytic polysaccharide monooxygenases. So slowly I started working on this type of an enzyme, and this Lytic polysaccharide monooxygenases, or LPMO, that's what we call, belong to multiple families. They all have the same structural fold, but they have very low sequence similarity among the different families. So the active site, if you see, it's actually quite planar, and this is from the bacteria and this is from the fungi, and they have the conservation of the active site residues, but the active site is very, very planar so that it can bind on the crystalline part. So it also has different types of mechanism, like type 1, type 2, type 3, depending on which carbon they attack, whether it's C1 or C4. And so our initial study was to find out what is the evolutionary pressure on these enzymes that causes some of these LPMOs to act on cellulose and others to act on chitin. And it so happens that there is an evolutionary relationship or the pattern that makes the active site and the residues that are part of the active site to be having an effect in the substrate specificity, especially when we look at even the carbohydrate binding modules that are associated with this LPMO. So again, those are static structures, and so again my interest went back, just like in endoglucanase, to see whether I can see any dynamics based changes. So again, we did the structural dynamic study with LPMOs using elastic network models, and Radhika was involved, Priya and Sheena were involved in this work. And so what you see here is basically the active site residues are red in color, and these are all the slowest modes, the first three slowest modes, which are orthogonal to each other. And you can see that there are certain regions, and especially just like in endoglucanase, the substrate binding site has more flexibility than compared to the non-substrate binding site. So this is the top view, and this is the side view of the same protein. And depending on which family we were looking at, we found the same pattern of dynamics between the different proteins. So we took the entire structural survey of the proteins and we did this study. So in conclusion about that particular part, we observed that the loop regions around the active site are more mobile. Their motions are in tune with the changes that it needs for it to bind to either cellulose or to chitin in an indirect way. But the residues that are predicted to be co-evolved are present on the non-substrate binding side of the LPMO, which I haven't shown the data yet. So at this point of time, I was very much interested to do something with Pulkit about a classifier, because over the period of time, new genomes that came up, new LPMOs got annotated. And there were some non-canonical LPMOs also that were labeled as being LPMO. So there was a need for some kind of an automated learning training or a model that can do functional classification of these LPMOs. So an existing method is dbcan, and the second version of that is dbcan2. It uses HMM-based profiles to identify not one family, but basically identify caseins. So endoglucanase, all these cellulases basically are carbohydrate active enzymes. So that's called as caseins. And there is a database called KZ, which actually annotates, and it's manually curated. It's a very good database maintained in France by Professor Henry Satt. And so what I did was, because for any learning, any classifier, you need to have really good amount of data. And so we took two families, AA9 and AA10. AA9 is from fungi and AA10 is from bacteria, to test the different methods. And so we used Protart, which is a library that generates about many different features, and each feature has many different descriptors. So in total, there is about like 9,000 descriptors in the Protart that can be generated for a sequence. And so the overall workflow was that we created a positive test set and a negative test set from KZ. We filtered the data and we got 60% of it for training and 40% for testing. And we did both neural network using these 6 significant features among all the features that we generated, and also an LSTM or a recurring neural network with a bidirectional LSTM to predict whether it's able to classify it or not. So in both the cases, what happened was, by the time we did this study, the KZ database had updated both the family A9 and A10 with new sequences that they had annotated as being belonging to that family. So what we did was, that was a very good data for us, because now we have an independent set which was like a couple of months down the line, that we could use it as independent set, and then we used it different parameters to test the quality of prediction. So just to go back a little bit, so in this there are 6 features that actually are significant, and the way we predicted whether a particular sequence is classified as belonging to A9 or A10 in the neural network was that we took an consensus-based prediction, meaning a common prediction. So let us say there are these set of sequences, and there are these set of features that we find, and in each sequence, if let us say a particular sequence has significance in this particular feature and in this feature and so on, we take only those sequences that have a significance being predicted from all those features as the LPMO. So it was, you can say, our filtering criteria to label a particular sequence was very stringent, and because we needed to reduce the number of false positives. So this is just to show what the LSTM network we had. The we had two layers of LSTM, both forward and backward, and here we gave up the output is giving up whether it belongs to A9 or A10. And so this is the data. This is the starting data for A9 and A10 with the positive set containing both training and validation. And we tried to keep it 1:1 ratio so that there's no bias. And we took those that are not A9 and A10 in the negative set, and then we found some interesting results. So we also used other training classifiers like stochastic gradient, SVM, using different kernels, and also neural network and LSTM. And also we compared it with DBCAN2. And here in DBCAN2, it uses consensus-based prediction as well and individual prediction. But for our sake of convenience, you can say, or again, to be stringent, we identified those that are LPMOs even by DBCAN2 if they all if hammer, diamond, and hot pep predicted them as LPMOs. So what you see here, the green cells are nothing but the F score values for the neural network that actually was pretty good for these both families A9 and A10. But at the same time, we do see some good F score for the LSTM method as well. So we have the codes and the model, the trained model in GitHub. This is Pulkit's GitHub with Predis LPMO. That's the name of the tool that we have kept. Please feel free to go to that and read the paper associated with it. With that, I think my time's up. So the initial part of the work with endoglucanases was with Dr. Tanner Sen. At the time, I was at Iowa State, and Tanner is now in Brain Genes in California. And then the later part of the work with the LPMOs, I did it with George Phillips, Professor George Phillips at UW-Madison, and then he moved to, and then also at Rice University. Most recently, the machine learning-based method, I worked with Professor Brian Fox and Eric Hegg at UW-Madison, Michigan State, respectively. And with that, I come back to the slide. So Yati and Shashank, they have been working. I forgot to update this slide. So Yati and Shashank have been working with creating a new knowledge base for LPMO in the last 6 months. In a couple of weeks, we are going to launch the website and have the knowledge base launched. And so quite looking forward to that. And Pulkit also has been helping in that same. So Shankar, he continued some of the thermostability based studies that we did, but not in bacteria, but in fungi. And we got some results, but they were still not so clear. And so, but we are still working on it. And with that, I thank my funding agencies and also the universities, Shastra University, and my collaborators who are closely associated with my work. And with that, I think I'll stop sharing. And if there's any questions, I'm more than happy to answer. Thank you.
Thank you so much. Yes, thank you so much for the great presentation, Raghu. I'm sure we all really appreciate it. I found it very fascinating. Like you mentioned, I'll open things up for a few minutes of Q&A if anyone has any questions. So I'll see if any stream in over the chat currently. I do have one for you. So do you have any insight into why the performance for the feature-based neural network didn't generalize to the test set for the AA10 family? Because when you were showing off the F scores, the F score for that family in the validation set was much lower than for the other families of networks and the other protein family.
Yeah, so the F score for the A10 family in the validation set was 0.9, and in the independent set was 0.7. So this was that could be. Oh, I'm still sharing the screen. Okay, okay. So what we think is happening in that particular part is either there are some features that we have missed in the new sequences that came up, because the sequence length of LPMOs usually is about 250 to 280 residues, but there had been few LPMOs that were annotated, which were like more than 1,000 residues in length. And so I think some of them were like maybe we didn't know about, or we didn't do the feature selection in those for those sequences. Maybe that was one reason that we think is highly likely happening. The other thing is that in neural network, what data set we used, these 6 features we found significant, not just individually but also as an ensemble feature selection. But maybe what would have happened is we have not optimized those features, because what PROTA does is that it gives you one feature, and that feature has multiple descriptors in it. There is no way to select the individual descriptors that have a significance. So if you have to select one feature, you take up both the descriptors that are performing good and descriptors that are not performing good as such as a whole. And that I think was also confounding to the effect of having the F score at 0.7 in the independent set. I think that's what we think is happening, but then maybe that is something else. I'm not sure.
Yeah. Well, that sounds like a great promising future direction for research to kind of better understand those features, both for model performance and to see if there's any kind of insight to be gleaned from the actual significance of all of the features. So it doesn't look like there are any other questions in the chat currently. So thank you so much again, Raghu, for coming to give your presentation. We all appreciate you taking time to share your insight with us.
No worries. Thank you so much for the opportunity. And if anyone wants to hit me with a question, I'm there on Twitter. I'm struckbioinfo, and do share your questions and any other queries with me. Thank you, Colin. Thank you, ISCB, and thank you to the Student Council for this. Thank you.
Certainly. Take care, Raghu.
Bye. Bye.